thank you, the Chemistry International webinar organizers, for giving me the opportunity. So I am, however, I'm not from the chemistry, and I generally do the physics. So I will try to really accommodate myself in the chemistry area. So the today what I am talking about is the fragmentation dynamics of camphor, and that is at higher photon energies. And this work has been done in collaboration with my students, and there are a few faculties involved abroad as well. So before going into the details of the talk, so this is where we are located in India. So this is the institute, Indian Institute of Technology, located at Hyderabad, and these are the signature buildings of the new institute. It's not very old institute. It's a very young institute, almost of the age of 13 years. So in this one, in Department of Physics, I have a lab wherein I have built many setup in last 10 years. I mean, since I joined, where one of the best the setup which I very first setup which we made was the velocity map imaging spectrometer, and this is what you will be hearing today about. And the even then little bit more powerful spectrometer we built was the reaction microscope. So, and the experiment which I will be talking about is the beam time experiment, which we have done in Electra 3ST. So today, so this overall flow of the talk is basically we'll start with what is the idea, what I want to really talk about. Then I will just introduce what the kind of experiments we do, and then we'll discuss the results. So really what our aim is to really look into the fragmentation dynamics here, fragmentation dynamics of the camphor molecule. So camphor molecule is quite bigger. I mean, the atomic mass, if the molecular mass, if you really look into it, is 152 atomic mass unit. So, and then when you look into the fragmentation dynamics, they can actually fragment into various channels. So can we really pick out one of the channels and really see that what is happening in that? So that is what our aim was. And here, the probe which we are using, generally when you are breaking it, you need a probe. So for that, the probe which we are using is the X-ray, the soft X-ray radiation. And when it really goes inside the molecule, it can actually, it can actually do many stuff in that. So it ejects the electron out, multiply charge, and then once the whole system is multiply charged, it can actually dissociate. So people have done, because this camphor molecule is a chiral molecule, so there is a mirror symmetry of the same molecule exists in nature. So that people have done photoelectron circular dichroism studies on this one. Even people are trying to look into the ion distribution and try to study the electron circular dichroism on that one. But what we wanted to do is basically we wanted to study the fragmentation dynamics, and we also wanted to add some environment to it and see that whether the fragmentation actually changes pattern or not. So we'll not look into that part, but we will look into the fragmentation dynamics of this camphor molecular ion. So to start with, what is this whole talk about? So we will be actually we will be shining a light in that. The energy range of the light which we are using is somewhere around 287 to 115 electron volt, which is basically is C1SH. If I say C1SH, means that there are different orbitals to the atoms, right? So the C1S is the closest one to the nucleus. So if you need really high energy to eject that electron out. So when you eject that electron out, so there is a possibility, the whole system is now very unstable. So there is a possibility that the electron from the top shell can go and really fill that gap up. So in that case, there can be a photon emission there, or it is possible that photon which goes out can be absorbed by another electron in the system, and then there is the electron will come out. So that process we call it as the OJT. Okay, so this ultimately leads to a multiply charged molecular ion, and then this molecular ion eventually breaks up. So this process from where the electron has gone out, that we call it as the OJ decay. So this is very fast process. and then the fragmentation happens, and then you see that the molecules are getting fragmented, and we are interested in looking at that. So this is the paper which we recently published, and most of those are taken from there. So how do we do this study? Overall study is basically done through the setup. This is a very special setup. So where we use the effusive jet. So there is a camphor molecules going in, and then through this skimmer there is a small hole, and then that goes inside the reaction chamber. And reaction chamber actually houses there are two spectrometer. One is the velocity map imaging spectrometer, which actually measures the momentum of the ions which are being detected by the detector. And the other side we have the simple spectrometer, which actually has just the MCP, microchannel plate, and it just detects either the electron or the ions which are coming from the interaction of the light beam and the molecule. So these two spectrometers which you see on top of each other, so they both can be either operated in ion mode. This means positive charge detection or the negative charge detection, and the bottom also can be operated in the similar way. So these detectors, what you see are on the top spectrometer, these are very special detectors. So though they not only detect the time of flight of the ions. What does that mean? Basically, when the interaction happens, the charged particle will be coming out. So the electron, the charged particle will move towards the detector. So the time which it takes from the interaction region to the detection region, that is called the time of flight. In addition to that, really the detector is like the photographic plate, you can imagine. So where it hits on the detector, you can figure out that where it hits. So you have a time of flight information, you have a position information. So once you know these three information, time of flight and the position information, you can detect the momentum ultimately. So when you detect the momentum of all the fragments which are originating, so then you can essentially have every information in your hand, and then you can essentially get the kinetic energy out. So so this is the procedure what is happening here. So let's say the photon comes in, there is a molecule, let's say AB, and then let's say the two electrons have gone out, and there is a leftover charge on the molecule is 2+. So, and because this is unstable, let's say this is unstable, so it will fragment into A+ and B+. I am able to measure A+ and B+ in coincidence. And when I plot the time of flight of A+ and B+, I will know that this molecule the AB has fragmented into A+ and B+ ion. And if let's say this is A++ and B+, then also I can easily figure out with the time of flight that the breakup has happened as A++ and B+. Okay. So so overall analysis actually involves you have the electron in your hand, you have the ion in your hand. Analyzing electron for the energy is much easier. However, analyzing ions as for the energy is little difficult, but we have actually established one technique which can give you the momentum of all the ions. So for the case of electron, so because this will focus the particular electron on the detect, know that particular energy has made up one circle. Okay, so because we know the photon energy, we know the atomic system which it has ionized. So then we can find out what is the electron energy corresponding to that. So that way we can calibrate our electron energy spectrum. But to calibrate the ion energy spectrum was little difficult task, because now in that case I am operating this velocity map imaging spectrometer in the ion mode, and I need to record the moment. What I am getting in my hand is the position information and the time of flight information. Okay, so then in that case I need to get the momentum also. So that involves, because this is not the homogeneous field, it was little trickier. So this is a lensing field. So we need to apply a lensing correction to that, so that I can get a correct momentum information. So this factor what you see here is F, is basically comes from the lensing factor, and this involves a lot of simulation. So just take my word that we with these factors which we can get out of the simulation, and that when you fit it in and you get the momentum information in there in your hand. So once you have the momentum information of all the fragment arising from one single breakup event, so I can find out the what is the angular distribution of those fragments from the which are breaking up from the parent molecular ion. Okay, so that this angular distribution I want to calculate. This is the momentum P1. What you see is the momentum distribution of first ion detected. P2 is the momentum distribution of the other momentum of the other ion. So then I know that what is the angle between these two ions. Okay, so that is what we wanted to we want to see. Okay, so then coming to the result part of it. So when we ionize, when we our camphor goes inside, and then we look into when it interacts with the X-ray, and we can vary the X-ray photon energy. Let's say we are moving from 280 to 305 electron volt. Okay, and those energies we are varying from 0.2 electron volt. So the spectra what you see on the left hand side is the ion yield spectra, wherein this what this site is basically coming from the ionization of the rest of the C atoms in the camphor molecule. However, this the peak at somewhere around 293 is basically comes from the ionization of the carbonyl atom. So there is a carbonyl atom, CO atom, CO molecule attached to that camphor. So that is where it is coming from. So what we will be actually fixing at, so we are actually putting fixing our energy at somewhere around 292.4, and then we will be doing most of the experiments here. What on this side, what you see is the mass spectrum. Now, mass spectrum is basically giving you all the kinds of fragments which are coming out. So this will show you whole jungle of fragment ions, because it's really quite big, huge molecule. It has a molecular atomic mass, molecular mass has 152 AMU. Okay, so now when looking at the photon electron energy spectra, so the black curve is basically from all the fragments. This is the energy spectra, electron energy spectra recorded for all the fragments which are coming from these fragments. And these those dots are basically coming from this. The blue one, what you see here, is basically coming from the 15, the electrons coming from the 15 AMU, and then the red one is coming from this red one you see here is coming from 25 to 31 atomic mass units. So these are basically for the all different fragments which are coming out. So these are the electrons from the fragments. Okay, so this is essentially telling you the binding energy, and we actually saw that with the Rene et al. So they have also published this this result long back, where they just look into the electron energy spectra, and which is matching quite well. The binding energies are matching quite well with them. So now coming to the main fragmentation channel, what we are interested in. So this is just a one-dimensional time of flight spectrum, and this is where our interest lies. So this is the very first, the third, fourth slide, where we saw that we are actually we are plotting a time of flight of first fragment versus the time of flight of the second fragment, and we are constraining ourselves to the ones where the first ejected particle is CO, which is neutral CO. Neutral CO we cannot detect. But all these fragments which are coming up here are basically corresponding to the add 28 to it. So that will be basically these two are the ions, 50 and 109 are the ions, and then so is the neutral. CO as a neutral going out. This as a neutral is going out, and the bond between this CO is quite strong. So CC bond actually breaks, and then CO as a neutral, it goes out. Okay, so then we are constraining ourselves only to those channels, not to others one, because there are a lot of further ions which are coming out, and we won't be. I mean, it is very difficult to analyze those fragments. So now looking at coming to the two-body breakup, where you see this one is basically is the two-body breakup. Okay, so in two-body breakup, the first mass is 55, mass 55, and the second is 97. This is the simplest channel, and we can essentially analyze. We can essentially say that when they are two-body breakup, any two-body breaks up, they will go back to back. Doesn't matter how or they are oriented in the space, but they will go back to back. This means the angle between these two ions will be 180 degrees. And this was the first result to really check it up, and then we saw that it is basically the angular distribution between these two ions is 180 degrees. And we also calculated the kinetic energy corresponding to that. And I mean, this is as of now is not in context with that. We want to look into the angular distribution. Now coming to the three-body breakup. So there are two three-body channels we are interested in to analyze. So there is in first there is a CS3 plus ion, and the second ion is C8H13 plus ion, and the CO neutral is going out. And the other channel, there is a CO neutral, as I said, will be going out, and these two are the ions which are going out. And what you see here in this picture is the angular distribution, where I have restricted myself that CO is going in one particular direction, and I am able to calculate, I am able to measure the angular distribution with respect to this. This we call it as the free body geometry. So I know now the distribution of C4. So I know, so this has to be noted. Sorry, this one should be. So this one is for the CS3 plus ion and C8S13. And here this is a distribution for C4H7 plus, and then this is the C5H9. Okay. So now looking at the first channel, where the first fragment is CS3 plus and the C8S13 plus ion. So this is the distribution, as I said. And here we did a simulation. We did molecular dynamic simulation, wherein we first optimized the geometry for the dication, and we looked into it that how actually it is fragmenting in time. So until and what we are doing it as that first, after the normalization of the optimization of the dicatonic geometry, we are increasing the temperature of the whole system. Increasing the temperature means we are basically exciting the molecular ion essentially. So initially we increase the temperature up to 1000 Kelvin, nothing happened. But around 25,000 Kelvin, what we saw that CO around 80 femtosecond CO as a neutral, it goes out, and after 290 femtosecond, we see that first CS3 plus ion, and the C8S13 plus ion actually they dissociate after a rearrangement of the hydrogen atom, proton rearrangement. So and when we look into this is for the case of so here when we looked into the simulated ejection of the angular ejection of the ions, and it matches quite well the angular distribution. The angle between these two and these two are roughly the same as what we were getting experimentally. So this is so what essentially is happening right after the breakup. Right after the CO goes out, the whole molecular system actually starts to rotate, and after 290 femtosecond, this molecule actually breaks up. And when we really make the line with the center of mass of the molecular ion, we see that the angles are matching quite well with the experimental data. And the similar thing we did for the other channel as well, wherein we increased the temperature further higher, so that nothing happened until the same channel we see until about 4000 Kelvin. But beyond 4000 Kelvin, we start to see that there are some other changes are happening. So CO actually ejects right after 50 femtosecond, and then these two molecular ions actually get ejected. And when we looked into the distribution, so the similar thing happens. So when we looked into the distribution and looked into the angular distribution of it, so the angular distribution matches quite well with the experimental data. Here also, right after the CO ejection, the whole molecular ion, the leftover fragment ion, rotates in space, and then after some time, after 250 femtosecond, it breaks, and then when you really join the angle, join the lines with the center of mass, we get these angles which are matching quite well with the experimental data. So, can we really conclude something on the temperature? So what we did, we really plotted the kinetic energy spectra for all the for the fragments, and when we deconvoluted the experimental resolution, apparatus resolution, and what what we fit was the Maxwell distribution curve, which essentially Maxwell-Boltzmann distribution curve essentially will tell you the internal temperature of the whole molecular ion, and that is what it tells you. So when we essentially deconvoluted that, so what we figured out that the temperature for for this fragmentation, so it matches pretty well. It is within 2000, 2500 to 4000 Kelvin. For this particular breakup, and beyond that, beyond that there is a different breakup. So basically, ultimately CO, which is being released, is carrying most of the energy, and when we look at that, the temperature, the curve fitting of the CO matches pretty well with our simulated data. So this means so our molecular dynamic simulation really correlates with our experimental data. So in conclusion, so we have utilized this, which was for the first time, we have utilized this momentum established technique from BMI PEPICO, and we have done this electron ion near C1s edge ionization. We found that we essentially looked into the dicatenate fragmentation channels, and our molecular dynamic simulations are actually matching quite well with our experimental data. Even the kinetic energy what we saw is matching quite well with the temperature which was simulated in the molecular dynamics simulation. However, I mean, to really decipher the dissociation route, we really need a detailed theoretical calculation. I mean, so far we don't have any theoretician in-house. Maybe in future we might be able to do furthermore. So this work could not have been possible without our team here, and most of the work is basically done by Mr. Sanket Sen, who is a PhD student in my group. And I would also like to thank all the funding agencies here, and also I would like to thank the organizers to give me the opportunity with this. I would like to